Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are very excited to bring you a new episode of the podcast. Before we dive into this one, we have a few announcements to make. The program of the upcoming third edition of the Dr. GPCR Summit is taking shape. This year, the meeting will be held between October 10th and 16th. The meeting will be virtual so that we can allow as many participants from around the world as possible. We will host live talks, which will be hosted on Zoom from October 10th to the 15th. Everyone is welcome to participate by either listening into the talks, presenting a poster, or submitting a pre-recorded talk. We will also have prizes for trainees for their great presentations. We are also trying out a few new things. We'll host networking and poster sessions for the first time on Wonder. We will have also a full day dedicated to trainees and to trainee talks. We'll be hosting also workshops on data analysis, on how to preserve data integrity in the lab, and how to use GPCRDB. Everyone is welcome to attend. And it's free when you become a Dr. GPCR Ecosystem site member, which is also free. Speaking of the ecosystem, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Visit drgpcr.com to explore the ecosystem. The ecosystem is your GPCR-focused virtual playground. Join over 50 of your peers who have already registered as site members. You'll also have the option to select a plan once you're a site member. You'll get access to all things Dr. GPCR, including access to new podcast episodes before they even get released to the general public. You'll get access to our new group discussion and forums, our job board, and our learning center, while you'll be able to take a course or even prepare and share a course of your own with your colleagues. You'll also be able to discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Once you're a site member, you can also become an ambassador, which means that you'll get your own dedicated Dr. GPCR Ecosystem affiliate link. And every time someone subscribes for the year to the ecosystem, you'll get compensated. This will potentially help you offset the cost of the yearly membership. Last but not least, we're also looking for content creators. Subscribe to the ecosystem and start writing your own GPCR-focused content, share it, and show off your GPCR talents. Visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the pleasure of having with me Dr. Nick Holliday. He is the CSO of Accelerate Bio. Hi, Nick. How are you?
Hi, Yamina. Nice to be here and to contribute to the podcast.
I'm so excited to talk to you today, and I know we've been planning this for quite some time now with all our schedule changes, so I'm excited that we finally get to chat.
Yeah, I mean, it's really good to be here, I guess so. And tell you a little bit about what I've been doing in terms of working for Accelerate and working at the university as well, having that sort of dual role.
Fantastic. Okay, so then let's start at the beginning. Would you please walk us through a short introduction about who you are and what you do?
Yeah, sure. So I'm Nick Holliday. So I've worked in GPCRs for a very long time. My hair's going a little bit grey now, I think, in terms of, and I forget that actually I started out and looked at my first GPCR way back in the mid-1990s in 1994. And since then, I've had a sort of academic career, and I'm now based at the University of Nottingham in the cell signaling group. Associated with Compare, so big centre for membrane receptors and proteins. So I work part-time there and have a research lab there as well, looking at particularly G protein coupled receptor molecular pharmacology. We're interested in all sorts of different types of questions and around signaling kinetics, around different types of mode of action at those receptors on that side of things. But then I took a step out a few years ago as well to actually join a newly founded company in Nottingham, and that's Accelerate Bioscience. And that's much more involved in drug discovery. So really what we do at Accelerate is to offer in vitro cellular and molecular pharmacology services to a variety of different clients. Some of that's on GPCRs, and we get really exciting insight into the way in which that field is developing in terms of drug discovery. I mean, it's quite an old field now, but one that's still really good to be involved in. But also on sort of non-GPCR targets as well. So it sort of broadened my experience, certainly, not only in drug discovery, but in different types of targets. But we'll probably stick to GPCRs given the nature of the podcast, I think, largely.
Absolutely. Although we love to talk about other targets, but it's difficult when the podcast is called the Dr. GPCR podcast.
No, understood, definitely.
So tell me, Nick, you mentioned that you're a pharmacologist, but I wanted to know how did you get involved in pharmacology? Were you interested as a young teenager in sciences? How did you get to studying GPCRs?
Yeah, I mean, I guess I think I was always going to be a scientist. I mean, I was thinking about that before I came on, and I know that really from the age of primary school age, which in the UK is sort of 5, 6, 7, I had this really drive to find out how things work. And actually, my dad was an engineer in the Air Force. So he used to go to all these conferences where they were selling jet engines to him, and he used to bring back these brochures and tell you how jet engines work. And I remember being really fascinated by that. My granddad was a farmer, actually, and he used to have a shed with bits of broken machinery. And my parents, when they used to want a little bit of time off, they used to basically put me in the shed and allow me to kind of play with pumps and motors and stuff that we found. It's probably not very good for health and safety nowadays, but that was the way I used to be entertained. And so I think in my early teenage years, I wanted to be an engineer. And then I think the teaching that I had and the biology teachers really got me excited in biology and neuroscience and physiology and studying the brain. And by the time I went to university, I think I knew I wanted to be probably an academic scientist. Even at undergraduate level, I knew that's where I wanted to go and do the sort of PhD and the postdoc route. When I was at university, actually, I did my final year in physiology. So I did one module, I think, in pharmacology. And actually, what inspired me was the switch to the PhD. So when I was doing my pharmacology undergrad, I met my future supervisor, Helen Cox. At the time, had just moved to London, was setting up her own lab, and was somebody who was, I think, in terms of sort of the collaboration side of the science and networking, the enthusiasm, was someone who really kind of made an impression on me, and so enthusiastic in terms of welcoming students to the lab and actually kind of making things work for them in terms of their studies. So that's really when the switch to pharmacology happened, because the project that I was involved with was actually on epithelial cells. So I actually started off doing electrophysiology and measuring fluid secretion in epithelial cell lines, but to particular G protein coupled receptors, to peptide GPCRs. And in fact, it was a very early sort of signature, if you like. I'm now interested in the kinetics of signaling, but actually way back then in the 1990s, we were looking at the kinetics of signaling of these receptors and thinking about things like receptor desensitization and things like that. So that's when my interest in pharmacology started growing. Over my years at King's, I did more and more in terms of the molecular side of things. So really understanding receptor mutations and studying recombinant systems and looking at receptor function at the protein level. And then I came up, obviously, to Nottingham around about 15, 16 years ago now, and joined, if you like, sort of a group of PIs that were led by Steve Hill. And I think that's really where my molecular pharmacology side of things took off, because now I was in a sort of specialist environment, if you like, where we had a group of people who all got on well, who were kind of really interested in similar sorts of questions, but sufficiently different to find your own niche in it. So I really enjoyed, developing my career that side. And as I say, I ended up with an independent research group there at Nottingham. But I guess one of the things that I also found was that, I was increasingly interested in actually the mechanics of how to do things, how to do assays, developing new assay technologies. We have a really good specialist sort of imaging-based approaches in Nottingham. A lot of the early work, for example, in fluorescent ligands on Brett and FRET, came out of the group as a whole. And my own perspective is that I wanted to use that in a way that's a little bit more applied, and I didn't really want to just write papers on it. And so, as I went through my academic career, I guess I was perhaps kind of thinking about, well, I'm really focused, I'm really specialist in doing this. I could do this as an academic for another 10 years, but what else is out there? And what else is possible? And around about 5-6 years ago, someone I'm now good friends with, Stephen Charlton, who was actually a lead at Novartis in the Horsham site, came up and became a professor at the University of Nottingham. And we got on a lot. We had similar kinds of ideas in terms of signaling kinetics, binding kinetics, but what Stephen really brought was the idea of how to apply that to drug discovery in that much broader perspective. And so that's really how I was, I was lucky to be offered this position at Accelerate, which Stephen and a colleague had initially founded, and to kind of start getting involved in that side of things and really trying to apply some of the pharmacology to answer some questions on compound profiling, for example, both for GPCRs and other targets. And that's how I ended up with this role as Chief Scientific Officer as well, leading that company.
It's so interesting that you mentioned your dad being an engineer and you being interested in applying assays and building assays. I think that's the engineer in you coming out and trying to use different pieces of a puzzle to answer a larger question.
Yeah, I think that's true. I mean, I think one of the things that it took me probably quite a while to realize, actually, and partly around sort of that dual role, is you get a feel of what type of scientist you are. And one of the things that perhaps when you start out, you don't realize there's a whole load of different types of scientists that are equally kind of involved in contributions and bring different aspects to a team, if you like. And for me, I guess, what I'm really good at is sort of the assay development side of things, the sort of getting the sort of pharmacological analysis, thinking of how to approach certain questions, particularly in drug discovery biology now. But I'm probably less of the person who kind of stands at the top and is the big extrovert and says, this is the next big idea and let's go and sell it for GPCRs. So my, I guess my role is partly in terms of being rigorous, having innovative ideas, but innovative ideas in terms of the assay technologies, rather than necessarily kind of thinking about how do we kind of do something completely different with GPCRs or answer a really big question. Like that's probably where I fit in, if I'm honest.
But I think you make a great point about knowing your strengths and what you like to do versus what you don't. Something, and I think it's important to highlight that because most likely trainees, and I'm speaking of experience, you get as a trainee, as a PhD or a postdoc, you get very specialized. And as an academic, you get trained to, formulate things as, the data suggests, which is fine. But at some point you need to acknowledge what you're really good at and also figure out what you're not good at, or maybe something you don't like to do. And then find the people in the team to complete that whole picture.
I think that's absolutely right. I mean, I think there's an element which I think some academic groups have as well now, which is really about team building, yes. Being supportive for early career researchers, and also kind of recognizing the different skills, because I think. In academia, certainly in the UK, for example, there are some aspects that are still overemphasized compared to people who are really, good, team players, for example. They contribute to the science that's going on, but perhaps, they don't get the sort of PI recognition. And I think in the UK that's changing. And I think actually, there are initiatives at Nottingham, for example, that are led by Jeanette and Steve Hill at Compare, that are kind of really trying to get that team science approach so that, the ECRs get due recognition for all the different things that they do. I mean, science wouldn't happen without the sort of postdocs or the PhD students in the lab. But I mean, one of the things that, I think is important for those the people starting out that I've learned in terms of the trainees is to kind of, figure out, what it is that you're good at, what are your strengths? How do you play to those strengths? And be really open-minded, I think, in terms of both in terms of the sort of subject areas, but also in terms of, kind of how you can use your skills, and the fact that, for example, working in industry as well as academia can be equally fruitful, and having an understanding of what's out there is really good. I mean, I know when I was a postdoc, for example, I was a postdoctoral fellow for about 8 years, and I probably got my fellowship at my last opportunity to do that. And I probably didn't have a plan B at that stage in terms of a career plan. So, kind of having that awareness that there's so many different ways in which you can use your science, and there are really fulfilling careers that are beyond just the sort of postdoc focused kind of particular subject area is something that's really worthwhile to instill at an early stage, I think.
I think so too. And I think with time, we're getting to that point where trainees are provided that freedom to look for that information, and that information has become more and more available, which wasn't the case 20 years ago. When I was growing up as a scientist, the thought was master's, PhD, postdoc, professor, nothing else. Mm-hmm. And we know now that it's not true.
Yeah, I mean, it was. I mean, as I say, it was similar for me as well. And I guess what I do see, I think, is the potential, at least, for more movement between different types of science. So I think, there was always the stage where actually, there was always the industry route and the academic route. And certainly, I think when I was, doing a PhD, if you made a decision to go into industry, you made it early, and I think there's still valid reasons for that, because there is a mentality associated with some of the objectives of industry that you need to learn early on and understand that well if you want to progress in that environment. Yeah. But it is, you can move between the two, and certainly going back from industry to academia, I think there's definitely options for people to now come back and go into lectureships in academia after having industry careers. And, I'm an example of, moving after being a middle-aged academic, moving into industry and doing something completely different, albeit with a foot in both camps as well. So yeah, definitely.
Agreed. I think you're a good example of that. And opportunities are more and more out there to balance out both and still use your skills as a scientist in both those worlds. I want to say potentially equally. And a lot of companies now have, postdoc programs. They sponsor postdocs, and then you spend that time working on a research project, but in an industry setting. So you get that experience as well, which I think is very valuable.
Yeah. I mean, I guess, if I was thinking about moving to one side or the other, I mean, I think one of the things that I love about academia still is the training and the teaching side. I think a lot of sort of, the support of the postdocs I have in the group, PhD students that I have in the group. I'm not sure how much they will kind of comment on the podcast afterwards, but it's something that is really important for me to see that they're successful and they do well and they get the best training that they have. And there's nothing better than seeing their first paper come out, or kind of seeing how they've got on and got postdocs elsewhere and things like that as well. But certainly, I mean, at Accelerate, we have quite a young team. We've got some people who are coming out of graduate or PhD level, really, really bright, really motivated, high quality individuals, and you get the same ability, if you like, to train and hopefully shape some of their careers as well at that side, as well as academia. So, and as I say, one of the advantages is we can host people, we can kind of bring people over from their PhDs or masters as well, to come and have a year and see what it's like in industry. Because one of the things that we do as a CRO, as a contract research organization, is we do a lot of different stuff. So there's a lot of exposure to different types of techniques and different questions that people have in drug discovery as we go through it.
It's a nice way to get exposed to many things and learn new technologies. So I wanted to ask you, do you remember when you first heard about GPCRs, and what was your impression?
Yeah, well, I guess so. Did I know there were GPCRs or not? I'm not. So I guess I did my PhD in 1994. That's when I started, and it was on the neuropeptide Y family. And actually still have some projects on NPY 20, 30 years later as well. But at that time, I think we knew something about GPCRs, but I think when I started my PhD, it was only just after the first NPY receptor had been cloned. So we're at the stage where actually we didn't know a lot about these receptors other than we were starting to get the DNA sequence and knew that they belonged to the GPCR family. So I think it was really right at the start of the PhD where I kind of began to understand sort of what these receptors were doing and the fact that they were involved in lots and lots of different processes. I guess before then we probably had I think I probably still saw a 7-transmembrane domain receptor diagram still in my undergraduates, but it was really early days for that. Probably the sort of G protein cycle was much fresher in people's memory, I think, and some of that was starting to come through in terms of Bob Lefkowitz's work and others as well. So I mean, we've come on a long way in 30 years, but ever since my PhD, I think that's probably where I've had a foot in GPCRs all the way through.
What was your how did it make you feel to know that it's a 7-transmembrane domain receptor and it does all of these wondrous it controls all of these physiological responses? Were you fascinated by it, or you're just so focused on the NPY receptor that that was your model receptor?
Yeah, I guess. Well, I mean, I think I was fascinated by the techniques that we could use to study it at the time. Mm-hmm. So I think there were at the time we started it was almost like there's elements of a black box. We didn't even we didn't have much of a crystal structure. I think we were going on sort of the idea that it might be like Nigel Baldwin's structure, which came out in the early 1990s for bacteriorhodopsin. So there's some information there. So it's some sort of bundle. And at the time, and a lot of the way in which we understood, binding sites or signaling was actually by changing the receptor protein. So I was really interested in terms of how we kind of get information from those types of receptor mutagenesis studies. Mm-hmm. And at the time, there was a lot of stuff that we didn't know about. We didn't know about, post-translation modifications, really. Phosphorylation was just starting to kick off. Arrestins were just starting to kick off. So there was a lot of, stuff to kind of come along and look at. And actually, one of the grants I got as my first postdoc with Helen was to look at palmitylation, which was, a fatty acid modification. At the time, we didn't really know what that did. We had a couple of examples for receptors, but nothing really in general for these GPCRs. So there's a lot of scope to understand at a fundamental level of how these receptors work. And then I guess, there was always I'm trying to think of when I sort of first understood that there were kind of 800 family members, but that was probably kind of relatively early stage. So I think, we saw that there was a potential for this receptor family, and the fact that we've got really, druggable targets within it, but we have a lot of scope to do more with that family overall.
It's so interesting the way you describe it, and you talk about methods and techniques to measure receptor function and all of these. Again, the engineer in you comes out definitely in there. So, I know this was the early days and, cloning and mutagenesis studies. Can you comment on how far along has the field come when it comes to methodologies to measure receptor function and to better understand receptors?
Yeah, I mean, I guess, I mean, it's chalk and cheese now, isn't it? Really, I think. I mean, if you look at the receptors that I like, which are primarily peptide receptors, chemokine receptors, and we have university projects on those, for example, at the moment. Even going back to the NPY family, obviously, one of the key things is really kind of having a sense of how ligands bind those receptors in terms of the structural information that's now available. We never believed that was possible. There's still an issue, I think, with peptide receptors, because if you try molecular dynamics with any sort of peptide that's more than 3 amino acids long, it's still quite open-minded in terms of how those things actually bind, and they're not always resolved in the crystal structures either. So there's still a lot to learn about the way in which the peptides activate the receptors. But we certainly have a lot more to go on, as well as there's a place for mutagenesis, but we've got a lot more to go on for there as well. We've got some fantastic, techniques in terms of being able to probe receptor function. A lot of the ones that we still use are sort of ones that we run in recombinant cell lines, but we can see now, and I think what will happen in the future is there'll be a lot more in terms of the way we can apply some of the things that we're doing in fluorescent ligand binding, for example, or signaling, in terms of trying to do that in native cells. So using CRISPR or modified receptors that way, or thinking about different ways in biosensors that probe that signaling. And that's something that we're really excited about, because with Stephen as well, I think we are always interested in kinetics and how kinetics impacts on function, because I think that is one of the key things that we can actually control with generating new compounds, new modalities, but also have an impact in actually how these drugs actually work in vivo and ultimately therapeutic as well. So, if I was going to pick, something that is emerging out of the GPCR field at the moment, which has actually been around for a while now, it's the sort of kinetics alongside the different modalities, the orthosteric, allosterics, that I think may actually make a difference, and is already making a difference to drug discovery at the moment.
Love it. You mentioned peptide binding receptors. Do you have a favorite GPCR?
Oh well, it's like picking your children, isn't it? I can't do that at all.
That's what people tell me.
Well, I guess my favourites change as they go through. I have to put a word in for the NPY family, because that's I've been with that for 20, 30 years, and they've gone through cycles. When I first started, there was a huge amount of excitement about these receptors because of their role in appetite and obesity. That cooled off, but now they're coming back in terms of other potential therapeutic areas as well. So that's always nice to see. If you stay around long enough, you come back into fashion, I think, probably.
Yep.
The one that I really like at the moment from our university work is a chemokine. It's CXCR2. Again, one that's not necessarily a very new drug target, but one of the things about CXCR2 is it was really one of the first receptors where we had an idea for these class of compounds called intracellular allosteric modulators. These are compounds that, for example, bind the receptor G protein coupling interface. And those to me are fascinating pharmacologically, because they immediately have potential, if you like, in terms of the types of allosteric effects they have, the way they act as antagonists, the way they may interact with different signaling pathways differently. And there's a whole scope there to actually sort of tailor compounds to give you a particular, if you like, outcome for the receptor that might solve some of the problems. And, CXCR2, the first allosteric modulator was again late 90s, early 2000, and a couple of companies developed those. I remember when that came out at the time, it was really shocking. We got this kind of molecule that's binding somewhere completely different from where the orthosteric binding site is. How on earth does that work? And one of the things that we're now following up is a little bit more in terms of the way in which those modulators work, but again, combining it with our assay methodologies to really understand more about that site in general for other classes of GPCRs as well. And that's a really fulfilling kind of avenue to be involved in. Overall, CXCR2 I think is a really interesting receptor as well. Just the basis of how chemokines work at it, the types of signaling it can do, the relationship, if you like, to immune disease and also oncology as well. There's lots of stuff to get your teeth into for that receptor.
I agreed. And I think chemokine receptors in general, that was my first contact with the GPCR field, working on CXCR4. And I spent the entire summer making dry mutants, mutating the dry motif, not using a PCR machine, believe it or not. It was site-directed mutagenesis using a phage.
methodology. Yeah, I'd see. Well, it kind of, we're from the same generation, M13 phage or whatever, isn't it? Single-stranded
exactly.
ones. I remember doing that sort of stuff. Yep, that's good. So, I always tell my PhD students they've never had it so good. They can now get it synthesized by gene arts,
Exactly.
and they have all the effort taken out and all the joy of kind of trying to get your colonies right. So
exactly. Oh no, definitely. And I think it was such a it was laborious, but it was still a challenge. And I remember joining the lab, and I was the first PhD. I was the first summer student, and the lab had just opened, and we didn't even have ATP, so we needed to find a buffer that contained some ATP for us to do our thing. But yes, using phage and making single-stranded DNA, and then what a fun fun time.
Well, I mean, CXCR4 is still popular, so you can go back there. You can dig out your dry motifs and see where it goes.
Yes, absolutely. I think there's still much work to do for CXCR2. Tell us a little bit more about its role in pathophysiology. You mentioned inflammation and immuno-oncology, but what do we know? What do we have to know about CXCR2?
Yeah, I mean, it's quite well established as a receptor target, I guess. I mean, probably the most familiar aspect of it is its role in the migration of neutrophils, and obviously there are a couple of IL-8 responsive receptors, and CXCR2, particularly in humans, seems to be the one that is relevant for it, and that means that it's been examined particularly in things like inflammatory lung disease, COPD, asthma, that kind of stuff as well, in terms of as a target to prevent inflammation, to prevent neutrophil migration into those sites. I guess one of the interesting things about it is when you think about GPCRs as drug targets, one of the key things to establish, first of all, is what else is that receptor going to do? What are the liabilities? What are the side effects? And I guess that's something where, I mean, for CXCR2, we've got very good small molecules. Some of them are the NAMs in terms of antagonism. But, I don't know kind of the status of all the clinical trials, but certainly some of them ended up with problems such as neutropenia. So the CXCR2 was involved in actually sort of white blood cell count as well as kind of the thing that you wanted. So and that's potentially where the question is, do we just junk that target entirely? Will we ever get past that problem? Because to get efficacy, we need to block CXCR2 in a certain way, and you'll do exactly the same in cells elsewhere in the body. Or can we actually get compounds that are able to discriminate between those two effects, whether it's through the way in which those compounds work at the receptor, how much they inhibit signaling, what pathways they inhibit, whether there's differences in the way the receptor works on different cell types as well. And, and that's kind of something which is, remains kind of interesting in terms of what we're doing in terms of looking at that receptor and looking at the modulators of that receptor as well.
That's a big question for many GPCRs when you try to connect them to a disease model. It's you want to have compounds that are specific not only to the GPCR, but in that disease state, and that are surgical to that signaling pathway or to that site, to that organ. And I think that there lies the difficulty. When it comes
I mean, that's something where I've actually, to be honest, learned a lot coming into the sort of more applied drug discovery environment in terms of thinking about that. And I think it's something that, again, if you give your early career researchers advice, it's like, all right, you want to kind of specialize for the rest of your life in GPCRs. But if you're thinking about that sort of application to drug discovery, be broad-minded, think about all the different issues that, if you're going to write in a grant, this receptor is really important for this disease, be objective about it. And, because it's very easy to take your pet GPCR and say, I've worked on epithelial secretion, so it's really good for an antidiarrheal drug. But then forget the fact that, well, if you make that drug, it's going to cause all sorts of other problems, blood pressure problems and things like that as well. And so that's never going to really make it in terms of an idea. So that kind of objectivity in terms of, drug discovery and thinking about GPCRs as drug targets, I think is really important. But you're right that, if you understand your objective about what the side effects and what might arise, then you can then start thinking about, well, how do we discriminate between them? Is there a way in which we can get compound selectivity, or the way in which the compounds work to give us selectivity in terms of a particular target tissue?
And which leads me to my next question, and I know the answer to the question is yes, but I would love to hear more. So, do you think GPCRs are still good drug targets?
Yeah.
The answer,
yes.
so far, everyone said yes, which is, the answer is yes, but.
Yeah, I mean, I guess, I can't reveal all the clients for Accelerate, but we have kind of we have a lot of people that are still interested in GPCRs as targets, and companies that have are investing in GPCRs as targets and doing some quite large programs associated with them. So, again, I guess it came in cycles. I mean, I remember sort of 10 years ago, again, people say, oh, GPCRs are sort of a little bit passé now, and people aren't quite so interested in them because they felt that they dealt with all the easy ones. But I think obviously what's changed a lot is that, there are new routes that we know to explore these more difficult receptors, like peptide receptors, for example, in terms of targeting with allosteric modulators, thinking about biologics as well as small molecules. Suddenly we've got a much bigger toolbox to kind of think about how we might work at these receptors. We know a lot in terms of genetics now, and I think one of the things that's really useful and provides really good evidence to start thinking about drug targets is where you've got a genetic linkage with a condition, because that is sort of unbiased in terms of being prevalent in a particular population, and you can make a really good case for particular receptors based on that as well. So I think they are coming back in fashion. We have, as I say. We've been lucky now. I mean, Accelerate's been going for about 5 or 6 years. We've got a couple of programs where the molecules are now advancing into clinical trials for the client. So, Accelerate is supporting the in vitro biology for that. We're not directing it, but we can see that some of these molecules are getting to the successful candidate nomination clinical trial stage. So I think the future's pretty bright still in terms of the receptors, and I think obviously the structural information, maybe some AI. We'll see how good that happens. We'll open up new ways to tackle them with good compounds, and I think that will kind of only get better as we go through.
I think so too, and I think you made a great point about the fact that a lot of companies have this renewed interest in GPCRs, and one thing that I've always deplored is that you go on any company's website and they talk about a field, immuno-oncology or immunology. And what is unclear from this side of where we're sitting is that what kind of targets. I understand that you can't reveal all your targets, but I have a feeling that GPCRs have always been targets within these companies, within these projects, but their focus is not getting a drug against GPCRs. The focus is getting a drug to treat a disease state. Mm-hmm. And whether it's a GPCR or another target, it is what it is. But I think please go ahead.
No, go ahead. That's fine.
I just wanted to say with the new methodologies, with the new technologies that allow us to, acquire data faster, analyze data faster, computational biology, as you mentioned, AI, ML, is also a new, renaissance of the GPCR field, with all of these ducks or all of these pieces that come together in order to answer questions. We don't need to make single-stranded DNA anymore to make mutants. We can just, order the construct and just move on.
Yeah. I mean, I think one of the things that's, that it helps a little bit with the kind of the wealth of information and the progress in technology, I think, is the speed with which you can make some of these decisions as well. So, it's now not like a 3-year GPCR program and then we decide it hasn't worked. I mean, you can actually get to a sort of reasonably good idea of, what the receptor does. Is it validated? Can we get molecules that are are patentable and actually sort of out there that kind of beat what's currently around, you can normally get a feel for that quite early on. So I think there's a lot of, it sort of de-risks, I guess, the progress, if you like, to evaluate these targets. You still have to come up for a really good case, and that's, a lot of the industry people that I collaborate with and they're just absolutely fantastic, and being absolutely objective and rigorous. They have to be, because there's a lot of money riding on it in terms of this is where we want to go, this is what's important to understand in the next 6 months, this is why we think the target is a good one, and that is really, really good. And I've learned a lot from that side of things, and those people who are doing that type of job.
I think so too. And I think, having the opportunity to work in a role similar to yours, where you have, you still have your active academic group, but you're also working in a company, it allows you to kind of balance that out, use your technical skills, use your analytical skills, the engineer in you, but also learn as to how others are doing it. And the priorities in a biotech or a pharma are completely different than in academia. Mm-hmm. But essentially, it still requires the same rigor, the same scientific rigor. I'm looking for the right word, but to be scientifically rigorous and being data driven. And milestone.
Yeah, I mean, it is. I mean, I, in a way, I get the best of both worlds. I don't always know which office I'm going to on a particular day, so that's a sign of old age potentially. But generally, that there's a lot of ability to look across the different sides and bring the best out of both academia and industry. I mean, I think again, one of the things that really impressed me is that I think in terms of technical quality, I would say that the industry scientists that we have at Accelerate, they are a step above kind of what we normally produce in academia at the equivalent stage, because what you often realize in terms of some of these projects is that the data quality is key. People have to make decisions on N of 1, N of 2, not N of 5 or N of 6.
Yeah.
So there's an element of speed there, and there's an element of reproducibility and having that rigor associated with it, which actually is kind of something that is important in academia as well, but you've got an N of 5 to demonstrate it, and probably a longer time frame as well. So there's those sides of things. That there's a side of that retentiveness that appeals to me in industry a little bit, in terms of kind of making sure we get really good quality data. On the academic side, there is a flow of ideas, and again, it's an example would be the fluorescent ligands that we now develop in academia, and the fact that those are really powerful tools to apply to industry, to understand binding affinity at particular sites, and to bring that into the elements that we do, which are sort of standard compound profiling, but also doing things like kinetic assays, understanding how long compounds bind for, that kind of stuff, and how that might make a difference. So yeah, there's lots of interaction between the two sides.
Which is amazing. And I always thought about, well, not always, but more recently, since at least I started to talk to people in different areas, both from the private sector and academia. I think it's two sides of the same coin. The priorities and the goals are different, but I think they're complementary, because you may spend 5 years developing fluorescent ligands or allosteric modulators, tools to uncover new things about new knowledge about GPCRs, and that can be taken into an industry setting where the goal is to develop therapeutics. But you've already taken the time to really get those basic answers to those questions in order to take that project or take that receptor or that methodology to the next step. And I think that's crucial to do. One doesn't go without the other.
Yeah. I think that's true. And I when I was early stage academic at Nottingham, I did a lot of collaboration with pharma, and we had shared PhD students and things like that as well. And that was a way of getting some of these technology ideas into industry and to kind of get them utilized a little bit more. And I think ultimately for me, that's kind of what's what I'd like. I mean, we can certainly write a nice paper on it in a pharmacology journal. That's where my research has generally ended up, rather than in Nature. but really what we want to do is to say, well, we've done this assay, we can understand a little bit about it. How do we actually then kind of use it or kind of apply it in a way that works for a drug discovery program? Because that's ultimately a little bit about what those assay technologies are about, as well as understanding a little bit more about how these receptors signal.
Exactly. We've talked a little bit about, giving advice to young scientists who want to contribute to the field, being open and doing what you love, acknowledging what you don't like to do or what you're not really good at, and collaborate with people who are good at those things. Any additional advice you would have for junior scientists who might want to contribute to the GPCR field or to science in general?
Yeah, I don't know. I mean, I think it's sometimes a bit difficult when you're starting out, but I think one of the things I've benefited from is some good mentors over the years. So I think I've talked about 3 of them. I talked about Helen, who was my PhD and postdoc supervisor. I took the idea of collaboration and networking out from her. She might not have recognized it, because it took me a while to actually go up and talk to people at conferences a little bit. But then I had Steve Hill, who's the head of group at Cell Signaling. Someone who gives really good advice, but also very objective. so he doesn't pull his punches. And I think you need that. You need someone to challenge you a little bit from time to time. And then, as I say, Stephen Charlton, who's, equivalent in age to me, I think, but came from a very different perspective and opened up a new area, if you like. And having those people through your career is something that's really important. And it's important if you're a PI, for example, to really try and support the younger generation. And that sometimes means that hopefully it means you put them first quite often in terms of the opportunities over what you do. And I think that's important. And yes, I think the open-minded side of things as well. I think, that there's an element, when I first started out in science, I used to read about everything, used to read lots of completely different things, but, yes, GPCRs are wonderful, and you get fantastic conferences. I've really enjoyed the conferences I've been to there, but every so often go to one that's much broader and go and step in a session that's kind of something completely different that you're interested in, because that sort of lateral thinking and feeding between different fields is actually really good, and something that works both ways as well, in terms of bringing different ideas in outside of your network, if you like, of lovely GPCR people.
I love that. And I think you make a great point about, stepping out of the field and looking at other, research topics and going to other conferences to see what others are working on. And if you're like me, if I'm doing something, I have to see what the utility is of that. And I think the utility could be not only to learn and to grow as a scientist, but also look at other methodologies that people are using to study other proteins. And think about, is it applicable to the field? Can we bring it in, and can we do something about using that methodology? For example, a lot of with I'm thinking about Brett and Fred. Those are great tools to study GPCRs, but those can be great tools to study other proteins. And it's becoming more and more abundant, where you can see Fred and Brett papers in completely different fields. But you could do the opposite as well.
Yeah, absolutely. There's a lot of opportunity there in terms of understanding the signaling. The kind of, if you look at sort of kinase drug discovery, for example, and you think about some of the things that are emerging now for, covalent inhibitors of GPCRs or slowly reversible inhibitors of GPCRs. The kinase field has been doing this for a while now. So, and certainly there's that sort of understanding, if you like, and there is that interplay anyway, but just learning from kind of, the same mechanism of action, but at different targets. There's a real way in which you can kind of think about those applications and what the risks are for your particular favorite receptor, I think.
Exactly. And one example comes to mind, Ben Myers, who's a professor in Utah. They study smoothened, and they were trying to figure out, found a protein that interacts with the receptor intracellularly, and they were looking at it, didn't know what to do. And then somebody said, "Oh, talk to this person," and that person is a PKA expert. And she looked at the sequence of the peptide and said, "Oh, it's PKA." And together, they, and you don't know. you're so familiar with your receptor. I'm sure you know some of the motifs or some of the amino acid sequences of CXCR2 or the NPY receptors because you're the expert at that. But we also have to acknowledge that we can't be experts at everything, and being able to branch out and talk to others can be beneficial to both sides.
Yeah, absolutely. I think it's rewarding as well. It keeps you fresh, and I think never stop learning as well, because that's kind of that's something that you can do it in academia through teaching and taking on some teaching where you're not expert in, or you can do it by looking at different fields and talking to people and understanding those types of things, and it just keeps you fresh as well, and you're not always kind of thinking about the same question for 10, 20 years, if you like, in terms of a particular aspect of receptor function you're interested in.
Exactly. Okay. I know we're almost out of time. I have two last questions. One, if it comes to mind, top three aha moments that shaped your career. I think one of those, while you think about it, would be having the opportunity to have this dual role. From what I'm hearing, it seems to me that it's an important aha moment.
Yeah, I mean, I think I would certainly pick that in terms of that sort of understanding of that opportunity to do something different, and to realize that I was about the right time in my career where I needed that, because it sort of reinvigorates you and gets you going for the next 10 years until we all retire. So I think definitely. I mean, I guess I haven't. It's an interesting question, because I haven't got the Brian Kabilka aha moments, I think, in terms of really revolutionizing the field as far as I can see. But actually, I think one of the things that you never get tired of is the first time an experiment comes off and it's a result that you predicted. And even if it's not going to shape the world as a new assay, that's fantastic. I mean, my last aha moment was probably yesterday. So we're making these fluorescent ligands for CXCR2, which are binding the intracellular site. And the PhD student involved has worked really hard in the chemistry, because it's not been easy. And we've got hints that these ligands are working really well. And then yesterday she took some beautiful confocal images where, surprisingly, because I thought these things would just be inside the cell, you've got beautiful specific labeling of the receptors. So just seeing that, and it's lucky when you're working in fluorescence, you get a lot of imaging to actually see the molecules that you're making. That's just fantastic. And it's going to give her a really nice paper, and it's something that will kind of really set her on her way. It's it'll be lovely, and people will like reading it, hopefully. But for me, I mean, kind of keeping hold of those types of moments, just in terms of the experiments, particularly when you can't do them yourself anymore, which is I've got to wait until I'm 70 before I'm allowed back in the lab, and even then I think people probably not allow me to hold a pipette. But that you never lose sight of the excitement of doing that kind of experiment, I think.
Agreed. And it's interesting you mentioned that people would most likely not allow you to get back into the lab. I think after a while, it happened to my PhD supervisor. When I started, we were working together in the lab because he just opened his lab. And I think after 2 years, he wanted to do something, and we said, no. Just tell us what you want. We'll do it. Because the comments were, I can't find anything. And, I think there was an incident when he did a PCR, but he forgot to close the PCR tubes. And because, after a while, you have to get out of the lab and let us do what we need to do.
Yeah, I mean, that sounds annoyingly familiar, I think. I mean, I guess we do a lot of 384 well format assays at Accelerate now, and I don't think I can even see the wells, let alone pet into them. So my clients can be reassured that I'm nowhere near the practicalities of the lab. We've got much better people to do that, I think. So.
All right, last question. If and when you have job openings, either in your academic group or at Accelerate, where can people find you?
Yeah, so Accelerate, we do recruit, and we're recruiting currently at various different levels. We have a website, which I think might go on associated with the pub site. That's acceleratebio.com, and I think you can find that. And the careers are there. We're also on LinkedIn as well, so we post opportunities there. But essentially, I think the thing to do is to write to us if you're interested. And I think we're always interested in coming to look at really good people to join our team. because we've got sort of fairly big ambitions, I think. So yeah, and I hope Nottingham's a lovely place to come in the UK as well. So feel free if you're international to come and think about us as well. On the university side, the University of Nottingham website has job opportunities. I think it might be a bit late for this podcast to go out, but we are actually recruiting for a lecturer in molecular pharmacology at the moment in the cell signaling group. The closing date is kind of coming up, but do have a look at that. So there's some permanent positions coming through, as well as all the postdocs that we have as a team go out on that University of Nottingham website, as well as jobs.ac.uk and sort of more general websites as well. So
perfect. And all of this information the links to these is going to be on your podcast episode page, so people can go there and find it from there.
Okay, fantastic. Well, thank you for that. That's very nice to have some free advertising at the end. So
of course,
always like to speak to good people. So.
Happy to do that. And with the new ecosystem where people can sign up, we will be starting up again accepting job descriptions. So there will be a form that you can submit, and then you can post your job descriptions there, so that people who are in the ecosystem or GPCR people who will be able to access those once they become a member.
Sure. Sounds good.
Thank you so much, Nick, for the great opportunity to talk to you. I really enjoyed our chat, and we'll talk soon then.
Yeah, thank you very much, Mina. It's been a pleasure talking to you and being part of the podcast.
Thank you, Nick. Thank you for joining us and listening in to this Dr. GPCR podcast episode. I'd like to thank our team members, Attila Forrest, Ines Pinero, and our newest Dr. GPCR protege, Montserrat Avila Zozoya. Welcome to the team, Monse. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at any time at hello@drgpcr.com. Until next time, stay safe.